Good morning, everyone. So I'm Natalie Piperidis, and it is my privilege to welcome you to the SRA webinar series. I'm very happy that I can see so many of you today joining us, and I'm confident that today's session will offer invaluable insight into the latest advancement of sugarcane genomic research, and that our must-attend event will offer important perspective and intriguing landscape of sugarcane research. I would like to begin by acknowledging the Aboriginal and Torres Strait Islander people of this nation. We acknowledge the traditional custodians of the lands on which SRA is located and where we conduct our business. We pay our respect to ancestors and elders, past and present. Today's webinar will commence with a presentation lasting approximately 45 minutes, followed by a 30-minute Q&A session. During the presentation, we kindly request that all participants keep their microphones muted to ensure clear audio. Questions will be entertained during the Q&A session, scheduled at the conclusion of the presentation. So without further delay, I am delighted to introduce our speaker, Dr. Adam Healy. I've had the privilege of attending many of Adam's presentations, and I must say, despite the complexity of the challenge working in sugarcane genomics, is always delivered impeccable presentation. I'd also like to take this opportunity to extend my congratulations and sincere thanks to Adam on the recent publication in the esteemed journal Nature. This publication revealed the complete sequence of the sugarcane cultivar R570, marking a truly remarkable achievement. It stands as a testament to Adam's great dedication and perseverance to this gigantic task. Adam works as a research scientist at the Genome Sequencing Center at the HudsonAlpha Institute for Biotechnology, a leading US research institute dedicated to advancing human welfare through genomics. His work as HudsonAlpha focused on developing genetic markers for complex polyploid plants and integrating genome resources to support large-scale pan-genomic agriculture research. But before moving back to the other side of the world, I just wanted to say to everyone that Adam is joining us from Toronto. But you know, like I think his brilliant career start because he earned his PhD from QAFI, the Queensland Alliance for Agriculture and Food Innovation, at the University of Queensland in Brisbane, where he conducted research on the genomic basis of desirable biofuel traits in eucalypt trees under the guidance of Professor Robert Henry. So without further delay, Adam, I invite you to take the virtual floor. And thanks again.
Thank you very much for those kind words. I appreciate it. Okay, how does that look to everyone?
Looks good.
Okay, perfect. Okay, well, thank you very much for the introduction. My name is Adam Healy. I'm a genome researcher at the Genome Sequencing Center at HudsonAlpha Institute for Biotechnology in Huntsville, Alabama. Currently, I work remotely in Toronto, and thank you very much for this opportunity to speak to you today. I'm very excited to be presenting to the people who will hopefully be using this R570 genome that myself and co-authors have spent so much time working on. It's also a little bit intimidating to be speaking to you today. I'm Canadian. I've only ever seen sugarcane growing in the wild twice in my life. Once was at a QAFI demonstration garden at the University of Queensland, and the other was driving north to the Daintree National Forest from Cairns some years ago. So before I get started, as I said, I work at the HudsonAlpha Institute for Biotechnology within the Genome Sequencing Center. It is co-chaired by Jane and Jeremy. they've been putting together, they've been at the Institute since 2008, and they've been building plant genomes for this whole time. We are a contract lab for the Joint Genome Institute, so we do a lot of bioenergy grass work and building plant reference genomes. There's a lot of expertise. They've been doing this for a long time, and there's certainly a lot of expertise in this group doing plant genomics. That being said, sugarcane has been the most complex genome that we have ever assembled. It's funny when you talk to people and you talk about the complexity of the genome, different things mean different words to different researchers. When you talk to a human researcher about it, they really get hung up on the size of the genome. You tell them that it's 10 gigabases in size, and they think that's, that must be so large. But the same information given to someone who does reptile genomics or pine sounds quite small. It doesn't have the most chromosomes. I think that belongs to ferns, and it's not the only interspecific hybrid crop that exists out there. But the breeders and the chromosome biology of sugarcane has really made things very complicated to sequence. This is a picture of the three first authors, myself, Olivier, and John. This is Plant and Animal Genomes 2018, when we were first creating these genomic resources. And it's probably too small to see on the paper there, but it was us drawing pictures of chromosomes and trying to figure out how things segregate and what we actually expect to see in the genome. So, what exactly about this genome makes it worthy of being published in such a prestigious journal such as Nature? So in order to answer that question, we need to sort of step back and understand the history of interspecific hybridization in sugarcane. I want to outline how the breeding in biology has generated the most complex genome that our group has assembled thus far. I'm sure there's more complicated things out there that we'll work on in the future, but so far this has been the most complex. I want to touch on why we need a new type of reference genome for sugarcane. So the complexity lends itself to needing a different kind, a representative genome assembly. And with that, I can tell you what we've learned from the genome and what we can do with it moving forward. So this all begins about 100 years ago. So there are lots of sugarcane breeding institutes around the world, but the one that I want to speak to you about is off the coast of Madagascar. It's a French colony called Reunion Island, where they do a lot of sugarcane breeding. Now, about 100 years ago, they were growing lots of noble cane, or Saccharum officinarum sugarcane, which produces lots of beautiful sugar in its stem. But about 100 years ago, a blight came through and wiped out most of the crops. And breeders realized that they really need to introduce environmental and disease resistance into their breeding lines. So what they started to do was to create these interspecific hybrids. So what they did was start making interspecific hybrids between domesticated sugarcane and wild sugarcane. This was the first step in creating that really complex genome. And the reason for that, you can quickly see what the problem is. So domesticated sugarcane, while it has many copies of each of its chromosomes, it's an octoploid. It's at least a stable octoploid. But when you start crossing it with wild sugarcane's ploidy varies wildly. It has anywhere from 5 to 16 copies of each of its chromosomes. So species can have 40 to 28 chromosomes. And it also has a different base number of chromosomes. Domesticated sugarcane has 10 base chromosomes, wild sugarcane has 8. But when you cross them, you can still create a hybrid between them. Now, these first hybrids, they didn't really have the sugar content that breeders were looking for. They had the disease resistance, but not the sugar qualities that they were looking for. So they started making these back crosses to reestablish sugar content. They would back cross back to domesticated sugarcane, and this is a process known as nobilization. So these new cultivars that were created, they had high sugar production and disease resistance as well. And those are the two central tenets that I want to sort of circle back to during my talk about what we've learned from the genome and touch on those topics again. So when you sequence the genome, you just sort of get a modern day snapshot of what it looks like. We sequenced the genome of R570, which is the world's most extensively researched sugarcane cultivar. But that just tells you what it looks like today. There's 100 years of history in breeding and biology that goes into this that created a genome that is highly polyploid. It's also aneuploid, meaning from one generation to the next, the chromosome copy number can change. It's a large 10 gigabase genome. It is interspecific recombinant, and it is simultaneously heterozygous and redundant at the same time. So what happened during this process of creating these cultivars? So it's sugarcane biology and breeding that has completely shaped the genome of modern cultivars. This has to do with how the chromosomes pair and how they're transmitted from one generation to the next, and that back crossing that breeders did in order to reestablish sugar production in those hybrid cultivars. These two things influenced everything from a genomic standpoint. It influences how we actually can construct chromosomes when we build the genome and how we represent them. It informs what sequencing technologies are actually informative for producing a genome sequence and annotation. It determines how well genetic maps will work when looking for QTL loci that underlie desirable traits. This one is quite important, and I want to circle back to this because I think this group will have a unique understanding of the challenges that QTL mapping happens when you try and do this in sugarcane. Lastly, once you have those candidate genes, how do you actually describe them and analyze them within an annotation framework? Once you have the genes, how do you adequately go through and determine what is influencing your trait of interest? So the first complex part of the genome is how the chromosomes pair during meiosis. So sugarcane hybrids undergo what is known as bivalent preferential pairing. So most of the chromosomes come together and pair during meiosis, but who they pair with can change. So to give you a visual representation of what that looks like, during meiosis, chromosome pairs start to come together. Some of them form strict pairs. You always find F pairing with a G. Some chromosomes between the two species are closely enough related that they can actually bind together, and you get interspecific hybrid chromosomes this way. But the two remaining groups, these chromosomes can change who they pair with. And this can lead to different combinations of chromosomes being passed down to the next generation, and also univalent chromosomes being passed down as well. So you can see here the chromosomes start to pair off, and who they pair off with can change depending on a number of factors. There's a paper that was published in 2004, Genome et al. And there's a single sentence in that paper that really summarizes why this makes things so complex in terms of assembling the genome. So they were looking at chromosome-specific markers that they genotyped in 282 selfed R570 offspring. And they found that about 65% of the offspring had different numbers of chromosomes than their parent. So it changes a lot from one generation to the next. This has a huge impact in how we represent the genome and what's useful for creating a reference that's going to be useful for this sugarcane community. The second aspect of genome biology that makes things very complicated is how the backcrossing occurs in sugarcane breeding populations. So as I said, the F1 hybrids that were created during the first interspecific crosses, they needed to be continually backcrossed to domesticated sugarcane in order to reestablish that sugar content. So in a traditional backcrossing scheme, you can see that in the first cross, when you make an F1 hybrid, 50% of the genome comes from one progenitor and 50% comes from the other. And then you continually do this backcrossing in order to reestablish, in order to change the ratio of the genomic component that comes from domesticated sugarcane. So you want a hybrid that has mostly the genome of the domesticated sugarcane, but also a little bit of the wild sugarcane in there for the disease resistance. Fortunately, when you start making crosses in sugarcane, that's not what happens. So when you start to make interspecific crosses in sugarcane, when you make an F1 hybrid, all of the genetic material, all of the chromosomes from domestication go into that F1 hybrid. So not only does more of that inbred domesticated material go in, the genome itself gets bigger. When you do this back cross again, the same thing happens again. only the F1 hybrid goes through the meiotic reduction and reduces, whereas domesticated sugarcane, again, contributes all of its chromosomes to the next generation, and the genome again gets bigger. So it's got more and more of that domesticated sequence within it. Now, this is where things really get interesting. This F2 hybrid is now genetically similar enough to domesticated sugarcane that during meiosis, things undergo the typical N+N reduction, and you get a more stable genome in the F3 hybrid. This has a profound effect on the genome itself because the genome size changes and there's more and more inbred identical haplotypes in the genome to sequence. This is great for breeders because it means that backcrossing is more effective in sugarcane than most other crops. That F2 hybrid, that first backcross, actually has 11% more domesticated sequence in it than you would expect with a traditional backcrossing scheme. Now, this was a nice toy example, and you could easily follow the chromosome transmission from one generation to the next, but that's not exactly how sugarcane breeding happens. There is a limited number of parents that are used in crosses. These elite cultivars tend to get used over and over again. And we see that in the R570 genome. So the cultivar POJ2878 was actually used twice as the maternal, as the double maternal and paternal grandparent of R570. And that means that about 12.5% of the genome is perfectly inbred. It's identical. It means that when you sequence it, you can't actually separate out the two haplotypes. So it's these reasons that the inbreeding and the backcrossing and how crosses are done transform the genome into the most complex one that we've ever attempted to sequence. So this is how you generate such a complex genome. I've modified one of the sentences from the paper to help highlight why this is a challenge. So sugarcane is an interspecific hybrid genome that combines all of the complexities that we see from inbred and outbred genomes that are on a continuum when the chromosomes pair between disomy and polysomy that have recombined, and they also experience aneuploidy. Now, when we build genomes, we try and come up with a representation that's going to be the most useful for people. So if it's an inbred grass, such as sorghum bicolor, that is inbred enough that we just typically give people one haplotype, one chromosome per meiotic pair, and that's enough to do genomic research with. But we can't do that for sugarcane because things don't pair predictably. If we were to generate a genome for something that is outbred, such as a poplar tree, typically that has enough heterozygosity that we can split things apart. The haplotypes split nicely, and we can give you both the genetic component that was contributed from the mother and the father. But that's the minority of the genome in R570, so we can't do that as well. And that weird chromosome transmission, 2N+N, in the backcrossing scheme has created perfect duplicate or triplicate copies of sequence in the genome. So that means that one piece of sequence in the genome assembly can actually represent multiple locations in the genome. We see this occur in other crops such as potato. Typically what people do is they artificially duplicate or triplicate that sequence, and they just put it into the location that they think it is most appropriate. But because sugarcane undergoes unique translocations and interspecific recombination, this prevents accurate placement of those sequences. So you don't know exactly where you should be putting them. So because of this complexity, we needed to come up with a custom assembly pipeline. And this was a major component of the genome paper that came out in March. So this assembly pipeline actually spans 3 separate generations of genome sequencing technologies. When we first started this, we had only Illumina short reads to start putting things together and make contigs. We shortly upgraded to PacBio consensus long read, which gave us some longer sequences and some a lot less contigs to work with when we started to put things together. And then we upgraded again when the PacBio consensus, high consensus hi-fi reads came out. These were so much longer and accurate that we rebuilt the genome again, and that's the version that you see today in the paper. In addition to these sequencing technologies, we also had 5 supporting genomic resources at our disposal. We had a bio-nano optical map that was put together by Corteva. We could leverage sorghum gene synteny when we were building chromosomes to make sure that things looked good based on gene collinearity. We had high-C contact maps, so we could look to make sure that our chromosomes looked good in that sense. We had a genetic map that we constructed, and we also had single flow sorted chromosomes. So collaborators in the Czechia and also in Australia flow sorted single chromosomes, and we sequenced those with Illumina, and we made unique genetic markers with those. So this is how we generated our primary genome assembly. This is our representation of the genome. So if you look at the 10 gigabase version of the genome that you're probably expecting, the 2N version of it, that is a duodecaploid. So it has 114 chromosomes, and its ploidy is roughly 12x of each chromosome, of each of its chromosomes. What we did was, because of the complexity and we wanted it to be useful for others, we actually represented only the unique haplotypes in the genome. We took identical haplotypes and collapsed them down and just represented them once. This version of the genome is about 5 gigabases in size. It's 67 chromosomes. On average, you will see 6 to 7 copies of each chromosome instead of 12. The contiguity of the assembly is really nice. It's 15 megabases. It's just a measure of how long the pieces are that go into each of the individual chromosomes. That means on average, each chromosome comes together in about 10 to 11 pieces, and there's hardly any gaps in the genome as well. If anyone is interested in discussing the nuts and bolts of how this was done, I'd be happy to discuss and take questions on it at the end. This is from the pipeline, though I'm always happy to talk about how we actually put this together. So that gave us our chromosome representation of sugarcane. We had the scaffold upon which that we could build biology. At this point, we didn't really know anything. There was just sort of this blank canvas of sequences that we needed to build a story out of. And of course, the most interesting things that we can talk about when it comes to sugarcane is what sequences are actually derived from wild sugarcane or domesticated sugarcane. Most importantly, which regions of the genome are inbred? Where do we think that there's collapse where we couldn't separate out those sequences because they're identical? And finally, where are the genes? This is what everyone wants to know. Where exactly are those sugar transporters that get switched on to put sucrose in the stems, or get switched on when a new pest or disease is going through the field? So we started by wanting to assign all the bases in the genome to coming from one progenitor or the other. So despite the fact that you can produce hybrids between these two different species, they've actually been diverging from one another for quite some time, about 1.5 million years. And during that time, they've accumulated their own specific repeat content. My collaborator and second author on the paper, Olivier Garcimur, from SRA, he actually extracted progenitor-specific repeats, and we used those to paint each one of our chromosomes. This worked very, very well. You can see here for chromosome 1A that when you take these repeats and you just put them on each chromosome, you can see the distinct blocks across each of the chromosomes of what was derived from which sequence. This worked really well for every single chromosome. We can do this across each of the different homologous copies to see where inversions have occurred, which progenitor they were responsible for, which pieces were collapsed and were uniquely represented, etc. When we look across each of the 67 chromosomes that were assembled, this is what it looks like. Here is all the primary chromosomes on the x-axis here, and each of the 10 chromosomes from sorghum bicolor on the y-axis here. So you can see any of the translocations that occurred, the fusions that have occurred, etc. We found that this chromosome painting to assigning things to a progenitor worked very well. We were able to assign 96% of bases to one species or the other. As we would expect from previous cytology, about 76% of the genome is derived from Saccharum officinarum, and 20% is from the wild spontaneum. But what I was really surprised about was how frequently interspecific recombination occurred. You frequently see chromosomes that start out as being domesticated, then they switch to wild, and then domesticated again. And you see this quite frequently across each of the different chromosomes, which was pretty amazing. We did a lot of secondary checking to make sure that this was actually true. I didn't put the wrong piece of sequence into the assembly. Now, this is the one aspect, this is, I hope, what will be the biggest takeaway for people, particularly for this group that I'm presenting to. The inbred regions of the genome are very important because they're why the genome was backcrossed and bred this particular way. Those inbred identical by descent regions can actually be identified and quantified. So what you do is once you have your genome assembly, you take those PacBio reads, those nice long pieces of reads, and you align them back to your assembly. We sequence the genome to about 30x coverage, and then you just look across each chromosome and you look to see where do you have roughly 30x coverage. That's considered a nice, unique sequence that was easy to distinguish. But we found frequently that when we looked across these chromosomes, you would see regions that the coverage would jump up to 90 and then 60 and then back up and down again. This is regions where the genome is so inbred that we cannot produce multiple contigs to represent it. All of that sequence gets collapsed down, and when you align to it, all the reads stack up at a higher depth than you would expect. When we took that information and intersected it with where each of the progenitor sequences came from, we found that roughly 86% of the spontaneum genome, or the contribution, was heterozygous and single copy. It always came out at the depth that we would expect mostly. But the interesting thing is that the domesticated sugarcane, those bases that came from Saccharum officinarum, only 48% of them were single copy. The majority of them are identical and collapse in the genome. Now, this has a profound effect on QTL mapping in sugarcane when you go looking for desirable traits and the loci that underlie them. And that's because the simplex markers that I'm sure most of you use when they're genotyping a population, the reason that you use a simplex marker is that they always segregate predictably in self progeny. So in this octoploid example, if you have a SNP that distinguishes one chromosome from all of the others, they're all the same. Regardless of how the chromosomes pair or how many copies they have, that simplex marker will always segregate 3 to 1 in a self population. But if there is a shared SNP, so a duplex marker, so such as a region such as this, where there's 2 copies of that region, in order to genotype that location, you would need a marker that would segregate 77 to 4 in a self population. Same thing goes for a triplex marker. That you would need 143 individuals to 1 in order to see that segregation. So it just becomes not cost effective to look at the genome using duplex and triplex markers. You need too many progeny and too many resources to actually genotype reliably across a genome of this size. What this means, though, is that when you're using simplex markers, based on these results, we were seeing that roughly half the genome is inaccessible to these markers. The other half, those simplex markers, are significantly skewed towards spontaneum. This is fantastic if you're looking for a trait that's related to environmental resistance or disease resistance. But when you're after the genes are being switched on that give you sucrose in the stems, these are going to be very, very difficult to identify. Now, it's all well and good for me to tell you that this is that these regions are more difficult to genotype, but I thought it'd be more appropriate to show you, just to show what the genome looks like from my perspective when we were trying to do this. So from our work when we were generating this genome assembly, we had a genetic map, and I was able to extract roughly 1.8 million simplex markers from that genome, from that genetic map. I took those markers and I projected them onto our assembly, and then I heat mapped the number of markers that hit any given position. So I just slid a window across each chromosome and said, how much of this sequence is covered by a simplex marker? So the multiple markers here. So you can see here, so this is chromosomes that have chromosome 8 homology. For each chromosome, I have painted it with coming from either efficient arm or spontaneum based on these red or blue lines down here at the bottom. And I've also provided that unique read mapping depth, how much of that sequence is actually collapsed down. And you can see here for regions that are not heterozygous, that are not single copy, there's almost no markers here that you can hit in the first 40 megabases of chromosome 8-10. Same thing here. You can see there's huge dead spots across the genome where there's just too much complexity or too much inbreeding to really distinguish what's happening in the genetic map. So if you want to start going after the sugar production genes, first you need an annotation. So this is something that came together really nicely. We took a lot of RNA-seq and iso-seq and from different labs. Robert Henry contributed some material, collaborators in France did as well. And we sequenced a lot of RNA-seq to annotate this genome. And we were able to annotate 194,000 genes roughly. 68% of the annotation is derived from efficient arm and 31% came from spontaneum. John Lovell, who is my colleague, he came up with this program that we call GeneSpace, where it looks at gene synteny or gene order across multiple genomes. So we can take what we have learned in other reference species, such as sorghum bicolor, and using that gene order, we can find the one-to-one orthologs in more complex polyploid species. So anything that is single copy here in sorghum bicolor from chromosome 1, we can look for roughly, on average, 4 copies in wild sugarcane. Oh, dead. In wild sugarcane, or 6 to 7 copies in R570. And I've also included the mosaic monoploid assembly that came out in 2018 for R570, just to show you how much more resolution we have here when we look at the genome annotation. So as I said, sugar content is the top breeding target for sugarcane. But these candidate gene regions are really, really difficult to explore in efficient arm because of their high copy number, and they're therefore more likely to be located in those inbred collapse regions where you can't use simplex markers. So we thought, well, once we have this genome, we could use comparative genomics from other species that we know more about and take a look at those orthologs in R570. So back in 2019, Cooper et al. Actually sequenced the genome of sorghum Rio. So unlike the genotype that is sequenced for sorghum bicolor, that's known as BTX623, that genotype is a serial genotype. It's short, it's early maturing, and produces grain. But Rio is a tall, late maturing plant that has high soluble sugar content in its stem. It's known as sweet sorghum. And this high sugar content is actually very similar to breeding targets in sugarcane. So what we did was we took the 49 candidate genes from their paper that they described these sucrose transport genes, hydrolases, transcription factors, and cell wall pectin synthases. We took that information and we looked for it in R570. We did that by first extracting one-to-one orthologs between sorghum Rio and BTX623. We then used GeneSpace to take the genes from sorghum bicolor and to project them into these other genomes and pull them back out again. So we knew the orthologs from R570, the monoploid path, and wild sugarcane. To look at the overall conservation of the genes themselves, we looked at total divergence. So we looked at the percentage identity relative to sorghum to see how much change had occurred in each of the different alleles. And we also wanted to see how conserved the individual motifs, the amino acids within the assemblies were, the annotations were as well for each one of these orthologs. Additionally, when you look just at the individual amino acids, that doesn't necessarily tell you if the chromosome, if the gene itself is functional. So in addition to that, we used a program called ESM-Fold to take each one of those protein sequences that we were looking at just at their secondary structure and actually fold them to see if any of the mutations that we saw could potentially impact protein structure. Could any of these mutations render things inoperable or knock them out? So this is an example of one of those candidate genes. So this is soluble sucrose transporter 4, or SUT4. This gene is actually deleted in Rio, and it's a candidate gene for sucrose partitioning and export. So if you look here, this is just a visual representation of that amino acid and all the different syntenic orthologs. If you look across this alignment, you can see that it is nearly conserved in every single position. There's only a few places in the peptide consensus that you actually see any difference between all these different allele copies in the monoploid path and sorghum. If we zoom in on one of those regions, we can actually see the actual amino acid changes that occur in each of the allele copies. So the SUT4 transporter on chromosome 9D has a that has changed to an S at position 389, and there's also a deletion at position 490. This is all, it's a really good example to show how much conservation occurs in these candidate genes and what they look like close up. But what people are really interested in is, where did each of these alleles come from? So what we did was we went through and intersected our progenitor bases with the locations of each of the genes. So we know for each allele where it was inherited from. So we have 4 copies that came from Saccharomyces fissionarum and 2 copies that came from wild sugarcane. We compared their protein identity, and we can see that this structure is very, very conserved. It's almost 98% identity across the entire 500 amino acids. We also wanted to take a look at the places where the genome was inbred and we couldn't represent all the different copies of each of those genes. So while we have annotated 6 alleles in the assembly, it actually represents 10 in total, because this Saccharomyces fissionarum copy is in an inbred region where there is 3x coverage. So we know that there's additional 2 identical copies in the genome that this annotation has captured. So you see 6 in the gene GFF, but it actually describes 10 in total. And we can also assign things a fold score to see how many high quality amino acids are in that protein that look like they fold well. So this is a very, very highly conserved sequence that looks like it's functional across all the different alleles in R570. But if we look at something a little bit more complex, such as glycoside hydrolase, or CYP2, this is a candidate gene from Rio that gets downregulated during initial vegetative growth, and then it gets upregulated later in other stages. And this has been validated in Arabidopsis, where CYP2 mutants actually accumulate fewer sugars in their stems. So this is a more complex alignment, but we can see regions of large deletion that occur between wild sugarcane and R570, between BTX623, between Rio, and all the different allele copies. When we focus in on one allele in particular, the one on 5B, we can actually see a frame shift mutation that occurs at position 744, which impacts everything downstream of that, the remaining 100 amino acids. We can actually see this change in the protein identity score. It's by far the lowest of all the different alleles that we've looked at, and the fold score is substantially lower as well. So it looks like this is potentially a knocked out allele in R570 that's not functional. Now that is sort of the first half of the story. Because we're talking about sugarcane, you have to talk about sugar transporters and sugar genes. But the other half of the coin is rust resistance or disease resistance that comes from spontaneum. I'd like to say that this work is entirely done by Angelique and Olivier. This is our collaborators' work, and we were fortunate enough to showcase it in this genome paper. It was a great story that we got to include in this work. But this is actually the first time that I've ever spoken about it because this was their work and I didn't want to talk about it until it was published. But our collaborators have been working on brown rust resistance in sugarcane for a long time. And that's because prior to 1980, brown rust around the world caused huge yield losses, up to 50% in some crops. R570 was actually the first resistant cultivar, and it enabled PCR screening for resistance. So there was a genomic loci that they could PCR, and if you had that sequence, you knew that you would be resistant to this brown rust. Even though researchers didn't know what was actually located at this locus, they knew that if you had it, you would be resistant. And that's because this locus is single copy and it's haplotype specific. But unfortunately, because it's haplotype specific, it doesn't recombine. So when our collaborators were trying to find map it, they could only get so close, and that lack of recombination prevented further mapping in the region. We actually have been looking at this sequence for quite some time once we had a long read assembly. This is contigs from the first PacBio assembly that we generated with consensus long reads. So as I said, the back clones and maps didn't cover this whole region due to lack of recombination. There was an unsized gap here. And Olivier sent me these sequences and said, can you find them in the assembly? And, is there any gaps in the region? And it took a very short amount of time once you had this assembly to do a quick alignment and then just dot plot, and found that this region was covered. It was covered by one contig and there were no gaps there. So this whole bit of sequence was filled in. I didn't realize at the time really what was found, but I could tell by the excitement of my collaborators that this was something that was very special and they had been working on for a long time. So the assembly and annotation of the genome actually filled this unsized gap and enabled our collaborators to actually inspect the candidate genes in this region that they had captured using back clones. So brown rust resistance is conferred by just two genes in this kinase pseudokinase complex that occurs in the tandem MRA. This is amazing to me because if you think about it, there are hundreds of thousands of genes in the sugarcane genome, and it takes just these two genes working in tandem that provide durable resistance to brown rust that has lasted for decades and works in multiple different regions. So another key consideration is that the resistance in the plant kingdom that works in this way is actually quite rare. There's only been a handful of instances where people have described it. I believe there's one in barley and another in wheat. So this is an opportunity to take something that we've learned in sugarcane genomics and apply it back to other crops. This has been something that's usually just happened in one direction. We take something from a simpler system and apply it to sugarcane. Here we can actually work backwards and take something that we've learned in such an important crop and look for it in other crops as well. So that covers most of the stories within the genome assembly that was published. But I wanted to touch briefly on the future work that should be enabled by this genome. So because of how we represented the genome with this primary assembly, we can use it to understand the haplotype or gene copy number variation that occurs in other cultivars. This becomes sort of our scaffold or our frame to frame questions and research of these other cultivars. And can we look for regions of high conservation where we could knock out all the different orthologs of a gene if we wanted to with CRISPR-Cas9? Or if we wanted to be more technical, could we knock out just the wild copies of an allele or just the domesticated alleles? This is just the start of work that this sort of thing enables. We can now start to actually go through and validate and test Brew1. We can find instances of other disease resistance loci, look at other cultivars to say, what happens if you have 2 or 3 copies of this Brew1 locus in your genome? What does that enable? The possibilities seem endless at this point, but maybe I'm an optimist. We can start designing new markers for non-simplex regions of the genome. So we can start to capture regions of the genome that traditionally have been lost when looking for domesticated traits in different cultivars. And also, importantly, and last but not least, this pan-genome sequencing network will enable us to sort of explore candidate genes from other model systems into R570. Sorghum bicolor was the example here, but we could easily extend this to Citeria, Miscanthus, Brachypodium, corn, or switchgrass, and look for candidate genes that we know well in other systems and look to see where they are in R570 and could they be described better there as well. This was a lot of work by a lot of fantastic people. I can't thank them all enough. It was great to have such a great group of collaborators to work with over the years. This work was driven by Angelique, Karen, and Robert. They submitted JGI community science proposals. That's how this was funded. These started back in 2017. It's spanned multiple years. And yeah, it's been a fantastic ride. If you have your ideas for other community science proposals that you would like to submit to the JGI, if you have a complex problem that you think genomics could solve, please reach out and we can chat about community science proposals. We do large-scale resequencing, pan-genomics, transcriptomics, functional genomics, so please don't hesitate to reach out and we can put you in touch with the right people to enable your science. And with that, I would be happy to answer any questions.
Well, thank you, Adam. That was like such a great talk. So now it's time for any question. So if you do have a question, you can type it in the chat or you can just raise your hand in using the hand button that's located at the bar on the bar at the top of the screen. And when prompted, just unmute your microphone and then mute again so we don't have any problems. So any question? Panday Gunja. Okay, sorry, I'm just going to read it. Wondering if chromosome painting for repeats was done in silica or with wet experiment.
The example that I showed there, that was done in silica. That was done bioinformatically. But then we took that information that we knew and we used cytogenetics to actually verify it. So this was a lot of work that Healy had done to make sure that the chromosomes and how we put things together actually made sense. And so everything sort of matched up nicely.
We got a question from Rahul. I'm just going to, Rahul, can you ask your question?
Hello.
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 Hi, Adam. Nice presentation and congratulations for published your paper in esteemed journal in Nature. I am just related to bioinformatics question. And recently we are also working on one low-decaploid genome. And one question: how to estimate the heterozygosity and ploidy level? Because we have tried many tools, GenomeScope and other tools to assess the heterozygosity as well as the ploidy level. And another one question, and you proceeded with the collapsed assembly to further, or you have used further, you have also used haplotype assembly to do further annotation. And
so to answer your second question, so the annotation was only done with the primary assembly. So that is the haplotype collapsed version of it. That is the best representation of the genome, and that's what people should be using moving forward. To answer your first question, how to assess the overall diversity and ploidy level of other cultivars, it works best if you have, once you have generated an assembly, if you can take something that is closely related, that is a diploid, and project the genes onto it, and then count the number of times you see really high quality alignments to it. That was the first thing that we did in order to take a look at the R570 ploidy. That paired with the haplotype collapse, the mapping of hi-fi reads back to the assembly itself, we would see a ploidy level of 6 or 7, but then with that additional information, we could actually look to see like, oh, okay, this is actually 10 or 12 copies of this sequence. So that is what I would recommend doing as well. If you look in the supplemental materials for the paper, it actually outlines sort of how the steps that we took in order to do this. And there should be some scripts there that should help facilitate. But if you have any additional questions, please don't hesitate to just email me and I can help out as well.
Thank you. Thank you, Adam.
Thanks, Raul. Now, Karen, you've got a question.
Hi, Adam. Can you hear me?
Hello.
Yep. That was a great talk. Thanks very much. You said you were talking about developing markers that have higher dosage. And we've given this a go quite a bit. And what we find is do amplicons in. The dosage isn't really related to the number of. Did you have some other idea where you other methods that you think that you could use for that?
Sorry, I didn't quite catch it. You said that the dosage.
So say you're looking in a region that's triple dose, and you amplify up an amplicon, PCR up an amplicon. We couldn't relate the number of reads to the dose of that marker, directly without looking at segregation, and so it doesn't always align. So it's all to do with how stuff goes up. So I'm just wondering if you have ideas to overcome that.
Yeah, that's a tricky question. I would say that I would probably try and if the reads or when you try and map things back, it's not easy to tell. I think probably a repeat masking to start with might be useful to make sure that you're getting good unique mappings. Okay. Yeah.
Okay. Yeah. Okay. Thanks.
No problem.
Raul, you still have your hands up. Do you have another question or was just from the last one?
I think last one.
Okay. That's good. Any other question for Adam? Well, actually, Adam, I've got one. So when you look at the BRU1, the sequence, the gene, so because of my experiments, I know that the BRU is on the chromosome 3 and it's on the official chromosome. So is the sequence like come from the spontaneous chromosomes or?
That is an excellent question. So this is something that we have actually looked at because I thought someone might ask it. I thought it was perplexing as well because that locus is smack dab in the middle of an efficient arm chromosome, which I thought was very strange. So we took those repeat markers and we just went, we looked across the entire chromosome at that haplotype. And it's interesting, there is good coverage of efficient arm markers on either side of it, but there is not a single repeat marker to assign it to spontaneum or officinarum in that region. Not one single repeat that can be assigned to one or the other. So I'm not exactly sure where it came from. It could be a very, very specific integration that we hadn't considered.
Yeah, I'm very intrigued because I was talking to some people in the States and they have like some spontaneum where they think that they have the BRU genes in a spontaneum. So I just want to have a look at it if they have it on each chromosome, because so far in all the clones that I looked at SRA, we've got one clones that have like 3 copy I've got 1 clones with 3 copy and 2 clones with 2 copy. Most of the clones at 1 or 0 copy. Then, you know, everyone think that like the spontaneum genome is supposed to bring all the resistance. So that's, you know, we expect it to be from the spontaneum, I suppose.
That was my expectation as well. But now that we've got this and other cultivars, we can hopefully do some comparative genomics to trace its lineage and find out what the effect of having multiple BRU1 genes or loci do for traits and resistance.
So did you have a look at any like the sequence? Do they compare to what's the synteny between for all the, you know, rust resistant genes that they found or they have in wheat or that sort of other cereal?
I didn't look at it quite that fine grain. It's sort of the same resistance motifs, but because the region is so small, it doesn't lend itself to synteny because you need multiple genes in order to find one of those syntenic positions, those ribbons that we see between the genomes.
Right. Okay. I mean, there's no other question, so I've got another question. I remember, sorry. I remember that I think in South Africa, they had found like another resistant source or gene for brown rust. And I'm just wondering, like, so that sequence, I know there is no other sequence such as the same one because there's only one site of BRU1 in R570, but is there any other part of the genome where they have something like similar, or you think that other gene that they found that supposedly, you know, have bring resistance is something similar, or?
It's possible. I haven't looked too fine grain into it, but we did characterize disease resistance motifs across R570. I think we included that as a supplemental table with the publication. And we found that there was a significant enrichment of loci that were derived from spontaneum. But the trouble with looking for those tandem kinases is that they're really difficult to find because they need to be in just the right orientation. You can't use just sequence homology because you'll find them, you know, everywhere, but it's not until you go through and really characterize the locus to say there's 2 right next to each other. They are oriented in the same way. One looks like it's a pseudokinase and is knocked out, but the other one looks functional. You need those combinations of things to actually say this is what is providing the disease resistance. And that's what makes it challenging to find. It's not straightforward as just saying, here is the ortholog, and here it is in the other sequence. It has to be more fine-grained than that.
Yeah. Yeah. Any other question? We still got a few minutes, and if you have urgent question to Adam. Adam, is that like the, sorry, is that like the, not worst genome, but the most complex of all genome that you worked on so far?
Yes, it is by far the most, even not just myself in the group, it is the most complex that we have completed. There might be some other things that we've taken a cursory look at that looked much more difficult. I can think of probably, I'm sure that there are auto polyploids out there with the same high ploidy that are very similar. That would be even more difficult to sequence. But this is by far the most challenging. It's also worth mentioning that before I worked on sugarcane, I actually, the last genome paper that I was on before this was for moss sphagnum. And sphagnum has 20 chromosomes and it's haploid. So it has the exact opposite problem of sugarcane. It has 1 copy of everything instead of 12. So it was different challenges, but much easier.
Yeah. Yeah. All right. Well, I just really want to thank you, Adam, for your excellent presentation. Honestly, such a complex, and I've heard some feedback already saying like you're really excellent at explaining like such a you know, something so complex, like very simply. And I think it's getting late for you. Maybe it's your bedtime. So I just want to thank everyone again for this, to attend to our session. I hope that you really enjoyed the webinar as I did today, and that you will join us for the next webinar. And you can see all this information on the events page at SRA website. The webinar will be actually put online later on when it's all worked out, and you can actually look at it again if you missed something. Thank you so much, Adam. Have a good night. We're going to go on with our day.
Have a good day.
Thank you.